Okay, thank for the organizers for inviting me to here and forcing me to listen to extremely enlightening lectures. I'm still trying to digest the last one. And also thank for the introduction. Now, I will try to describe an approach which is not necessarily opposite to what this complexity is about, but it give it some different angle. And also, I keep thinking about the issue of am I dealing with complexity or with simplicity? I will argue that in our field, things are quite under control and that we could do prediction. I think the last lecturer argues that prediction is too ambitious. Also in the Bible, they say that after the destruction of the second temple, the prediction was given to children and to fools, but we will see what we could do about it. So just to introduce you to the whole issue of complexity, in the Nobel week, there was this event called Nobel Mind, where they brought different people from the Laureate, and they talk about what they did. And the interviewer tried to steer a discussion between Fima to Schuller, where, as you know, they have different opinions. And Schuller was trying to convince Fama that Fama could predict what will happen to the world market, and Fima says that he cannot, and then Schuller say you can because you are very rich from your prediction. I think it's still a case of complexity where it's still very hard to make prediction in economy. Another example, which also is related to the last lectures, in 1983, we've been in the Pontifical Academy, and the Pope John Paul tried to tell the people in the meeting that, well, science has validity in some cases, but it will never be able to explain the brain or to deal with emotion. And at present, he is right, but we will see what will happen in the future. So what I will try to do now is to show or to argue that in many biological systems, the butterfly effect does not exist, at least the way I see it. So we do have complicated system in biology. This is just part, cell wall, photosynthetic device. We have light. We have motors that I will discuss. We have proton transport. Looks really, really terrible and very complex. I could go to another level. If you look on the cell, there are extremely complicated map of transaction of signals in the cell, of how something here activate the nucleus. And many of these junctions are proteins whose structure is known. And the question is, could you understand this whole system, or as a start, just look at part of it and try to see how they work? And one interesting and challenging part is this molecule called ATPase, which I will discuss and try to explain to you how does it work, regardless of your background. It's combined motion of protons from one side of the membrane to the other. This leads to rotation of a central stack. When it rotates, the cell produces energy by moving a molecule called ADP to a molecule called ATP. And essentially, I have a very nice video. It's not mine. It's from Creationist. You could go to Google and find it, who argues that this machine must have been invented by intelligent design, that there is no way that evolution designed it. So you could make this argument. You could also try to see how it works. Now, this is something that you do. Let's see how the movies work here. When you look on this machine, it's basically a pump activated by water. You do understand how it works. You want to reach a similar level of understanding when you go to one of these biological molecules. And before I go to different systems, I'll just show you the systems that we will consider. In the body, there are ion channels. These are proteins that conduct ions. They exist in all of these systems. They control neuron transmission and so on. The structure is known, and you want to be able to understand and to predict, using the structure, how these molecules are working. So it's a rational approach, physically based, trying to see how different biological systems work. And there is a system that I will not discuss, though we work on it. This was the Nobel Prize a year before last year. And I show it. This is what is called GPCR. It's when you have hormones binds to a molecule. This stuff could also be light, and what you will have will be rhodopsins that I will show you. And this activate cascade of events that lead to different reactions. They could activate metabolic pathway. They could send signal to the brain. And I put it just listening to the previous lectures, because there would be people who would argue that this is the butterfly effect, that this guy, when it binds here, somehow activates the rest of the system. And since we do not understand how it works, it must be completely chaotic, I would say, voodoo type of activation. And I will go in the opposite direction, try to show you that at least in this type of systems, you could build models that will predict and explain how things are working. And I will, of course, be happy to entertain opposite opinions. There are more systems, and let's start to go to examples. One of the systems that I will try to concentrate on, maybe the first half of the lecture, would be enzymes. Now, here we start to be slow. Not everybody knows what are enzymes. Enzymes are proteins that do chemical reactions much faster than what happens when they are not around. You could see here, again, this is the enzyme. We are trying to break this bond. When you put this system in a test tube, it could take months until the reaction occurs. When you place the enzyme in, it happens much faster, sometimes 20 order of magnitude faster. And one of the key questions in biology is why it is faster. And also, I recommend for philosophers to follow how this question was distorted by people who don't know to answer it, how it was trivialized. Well, in many cases, the answer was that it is faster because it is faster. So these are very serious people. So we'll try to see how or why they are faster, and to contradict it with approaches that don't use physical models. OK, so now we want to model molecules. This is a part of a protein. There is a main chain, and there are side chains, which are different groups. Some of us know them, some don't. But this is just a molecule, connection of atoms in a chain. In the case of a protein, they fold to a unique form that allow it to do what it's designed to do. And I will also discuss the folding process, showing that it's not, some completely chaotic process, and try to explain why. Now, we know a lot about proteins. Enzymes were discovered more than 100 years ago. Eventually, people realized that they are proteins. The first enzymes was solved by D.C. Phillips. This was lysozyme. And shortly after, David Blow solved the structure of chymotrypsin. Max Perutz and John Kendrew solved the structure of proteins even before. And during the year, there is a huge accumulation of structural information. So we know a lot about the structure of proteins. There is a whole field of protein structural determination. This is an earlier slide, but it shows you that we know the structure of almost any interesting biological complex, including membrane proteins. Now, knowing the structure is essentially not enough to understand how a protein works. The example is if you see this watch, you could see the watch, you could even see what it has inside, but most of us would not be able to realize how it works just by looking at the watch. Now, the analogy is that I would say that biochemistry is the discovery of this clock. Crystallography shows us all the parts. Spectroscopy, including single molecules that I will consider later, could show you how fast these wheels are moving, but we still do not understand how it works. And understanding, I mean being able to predict how fast different proteins are working. So here I make a junction. I claim very loudly that you cannot understand how proteins are working without computers. That is just too complicated for the human brain to look at the structure and to figure out what are the forces inside the protein. And you need a computer to kind of extend your brain. I don't know how it would be classified in philosophy, but to me, this is a fact. This is, you see, you need computers. This is the computer we used the first, the Golem, which come on the name of this magical robot that the rabbi from Prague built to do his work. This was second computer at the Weizmann Institute, and it was very powerful at its time. So I argue and will show you that you need computer to understand, but then you have to convert what you learn by the computer to conceptual picture, which require the brain. So let's go step by step and show you how you try to understand or model an enzyme. So first comes the part which is relatively easy. This is for my books that you are welcome to buy, where this is a part of a protein, and you want to model it. Now, there are different levels of modeling. If you just want to understand the structure, you could make major simplification, which are, I will argue, completely valid. What you do, you take each of these fragments, like this part, and we will agree for a minute, though we heard a lot of bad things about the complexity of quantum mechanics. When you go to quantum chemistry and study the energy of stretching a bond, you get, with high enough level, you get the exact result. Quantum chemistry for small molecule is a done science. It gives you the correct observed results. So if you look on the bonds, you could get the energy of stretching a bond. And since we are not going to break the bonds in these next 2 minutes, you could represent the bond by a spring, where the force constant for the spring agree with the experimental or theoretical strengths of this bond. You could use the same approach for bending angle. You could use similar approach for interaction between atoms which are far away. And you will end up with what is called force field, which is mistakenly called also by the Nobel Committee classical mechanics. It's actually what is called molecular mechanics. You replace the forces by considering bunch of ball and springs. And this approach, just a minute, works extremely well to describe the structure of molecules, the vibration of molecules near the minimum, and there are countless simulation programs, all of them originated from the same program. These are some of the original work which was done with Sneor Lifson and Mike Levitt. You could describe the motion and conformation of large molecules by these ball and spring models. And you could also try, if you have a large enough computer, to model the structure and how it changes with time. This still does not allow you to ask questions about how enzymes work, about chemistry. You have to do something more. So we agree temporarily that molecular mechanics allow you to describe structures. You want to be able to describe function in chemistry, so you want to be able to break bonds. And the way to do it is what become to be known as QMMM, combining quantum mechanics for where you are breaking bonds and molecular mechanics to where you just change structure. Now, originally the molecular mechanics, quantum mechanics, molecular mechanics start with these two work, which treated separately the quantum chemistry and the rest of the molecule, which was described by molecular mechanics. This approach could not really tell you how protein is working because it misses the main hidden link in this, which is the coupling between the surrounding to the quantum mechanics. I will be more specific. When we start to use this approach to study the first enzyme that was solved by D.C. Phillips, lysozyme, and we have separate molecular mechanics and quantum mechanics, we found that we cannot break bonds because the energy of breaking the bond would mean that the bond will never break in reality. It was just too high. And after thinking about it for a long time, we realized that what would make this model workable is to tell the bond when it is being broken that there is environment around it. There is solvent or protein where when you break the bond to plus and minuses, the environment stabilizes this plus and minus. So here it's one example where you don't really, where you try some approach, it does not work, you think about it for a while, and suddenly you find what you missed. And I would argue that if you do this repeatedly, and you reach a stage where your predictions are correct all the time, probably you found the missing link. In this case, I'm sure, so I will show you again. When you break a bond, and you don't include the surrounding, the bond break with what we chemists call biradical. It takes a lot of energy to break it. When you include the surrounding, suddenly the bond tend to break as plus and minus with much lower energy, and this coupling is the key to what is called QMMM. You couple the molecular mechanics electrostatic with the quantum mechanics, and you get a workable model. Now, this type of approach is it's a general idea of what is called now multi-scale modeling, where you focus on the important part and describe the rest on a lower level. Now again, people who believe in long-range effects, and I will show you again and again that long-range effects are well understood, will say that this is wrong because something here could make this cat do meow. It's essentially incorrect. If you have a methyl group very far away from the active site, it does not do anything. Now a point that all of you will agree, but it's good for other people, that if you try to model something, you have to model the totality. It is not enough to do very well in describing part. What happened here? Okay, it was supposed to show you that you see that you know exactly how the chairs are built, you know how the window is built, but it does not allow you to design the airplane. So you do have to model everything, and the question comes, who are you to know to model everything? And I will come to it again and again. So you want to model the totality, and you also want to do things in a simple way. And the most powerful approach at present to model enzymes appear to be something that I learned in physics, which is based on saying that I will model best the asymptotes, the reactants and product, and this will allow me to have quite a good idea on what happened at the reacting region. So this approach is called the EV beam. I will show you here, empirical valence bond. And what you do, and again, I admit that there are people here who don't know chemistry or know high school chemistry. This approach is basically taking high school chemistry and realizing is that it is the best way to do biology. And it goes like this. I want to model this reaction where this negative CO minus attack carbon and send Cl minus far away. What you do, you have a very nice force field, this ball and spring which describe the reactant. You have a very nice force field that describe the product. And then you mix them by a mixture term and require that the mixture will behave as the correct energy of this system behave. And the correct one is what you obtain from doing quantum mechanics in some isolated system. So you basically mix two simple representations, force the mixture to reproduce reality, and then come the tricks that makes it reliable. You fit this stuff to what happened to this reacting system in solution, and then the only change that you do when you move to the enzyme, you replace the solution by the enzyme. So this approach does not try to discover quantum mechanics from the beginning. It looks on the change in this profile when you move from water to the protein. So the way we ask how enzymes work is we look on what is the change when you move from having this system in water to moving to a protein. And I will show you how well it works. Before, again, I keep going back to some simple point. If you want to study reaction, and you know the energy as function of going from reactant to product, you should use this law, that the rate constant is proportional to the activation free energy, plus some factor, which in condensed phase is about 6×10 to the 12. Now, at the early time when Eyring and others suggested these formulas, the physicists did not like it. This is probably why Eyring did not receive the Nobel Prize. But during the year, it appears that this is essentially exact when you add a transmission factor, which tell you what happened to a trajectory that reached this point. If it decides to move twice back and forth, the rate constant will be half of what it is. But what determines biology, chemistry, and so on is actually the activation free energy, this height. And I will, of course, present the opposite point and show that it has no merit. So let's agree that this is what we want to find, which means that most of the physics that we will be exploring is what determines the height of the barrier. Of course, we will allow people who don't like it to present their point of view, and then we show that they are wrong. So if you want to understand enzyme catalysis, the most logical thing is to say that in water, the reaction is very slow. In the enzyme, because of something, this barrier is being pushed down. And the question reduced to why or what is about biology that push this barrier down. The previous lecture talked about presenting the question. This is the question in catalysis. And when people say that they don't believe it and there are other effects, I show who is this guy? Yes, so Boltzmann essentially was right that free energy is what determines everything, and I will show you several examples where this works. So I will start from examples that go directly to biology, show you what we do, and then I will go to several consideration of what could be other reasons for catalysis. Okay, so this is a complicated cycle of an enzyme called RAS, which take a molecule which is called GTP. These are 3 phosphates, 3 negatively charged phosphate. And when a protein called GAP binds to this RAS, this GTP is broken to GDP, which are 2 phosphate, and to isolated phosphate. And now why it is so important? Because if there are some defects in this RAS, some side chains are incorrect, you will get cancer because this bond will not break and the cell will continue to differentiate. So it's kind of example of important question. You want to understand why and to also predict what different mutations will do. So, yes, I gave here a lecture in July and the color was bad, and now it's again the same story. So this is supposed to be this GTP, which you don't see clearly. This is a residue that when it mutated, you get cancer, and you want to understand how it works. In order to understand it, you have to first guess or have intelligent idea of what is the chemistry. Then I will show you how you get more intelligent guess. And then to try to use this EVB and to say how the enzyme changed this barrier. Now, if you are just experimentalist, I say just, you will not know, this essentially is my postulate, you will not know how the reaction occurred. There is no experiment, despite 50,000 people who work on it, there is no experiment that actually tell you how this phosphate is being broken. Because the barrier is so high that there is no direct way to go there. The way to know the reality is to run quantum chemistry programs, to spend several years to learn about the system, and then you find that this is the free energy surface that tell you how the reaction occurred. And during the year, you improve your results, but quantum chemistry is really the only way to know how the reaction occurs, not in the enzyme, without the enzyme. And I contend, though probably there are very few chemists here, that this issue was never resolved. Essentially, I wrote 200-page recent review why nature really used phosphate, where I made the point that this could not be figured out experimentally. Anyhow, after you figure out what happened in solution, you develop this EVB and run it in the protein. Then you get this nice movie where this is magnesium. I will show you again. The water molecule attack the phosphate, and this bond is being broken. So you could produce nice movie of the motion of the atoms. Maybe I'll show you again. This is a water molecule that's supposed to attack the phosphate. And it does, and it breaks the bond. But this is not what we want to know. What you actually want to know is what is the energy of breaking this bond in water, in the protein, and in bad proteins where you have some mutations. And our approach allows us to calculate this quite reliably. And essentially, if we go to other proteins, we could do mutations on the computer, change residues in the protein, and figure out how the rate of the reaction change in the protein. And this is example. This is a protein called charismat mutase. These are different forms of the protein, different mutations. And one of them, the blue, is the barriers that we calculate. The black is what is observed. So we have the ability, arguably, to calculate the speed of the reaction in different proteins And we could do it in many proteins. These are a series of proteins where we actually reproduce the difference between the reaction in water, the reaction in the protein. And we are doing quite well in the last 30, 40 years. Now, the first interesting point is that in all of these cases, we found out that the reduction of the barrier is due to electrostatic effect. In a minute, I will tell you what type of electrostatic effect, but it's not due to different factors that I will show you soon. It's due to having the right charges in the active site so that the transition state is being stabilized. Now, this brings us to the point that there are other people who would tell you other ways. For example, many years ago, who is this? Yes. This is Linus Pauling in a famous lecture where he presumably explained how enzyme work. This was before there was any structure of enzyme. And basically what he says is that the enzyme changes the activation barrier, which is correct. But he could not tell of how does it change the activation barrier. This was the question. And a lot of the people who refuse to understand how enzyme work will say that it was explained by Pauling because he says that enzyme reduce the barrier. As I told you, the question is how. So there are about 25 explanations how enzyme could work. And 24 of them are incorrect. We will argue later with the mathematician of how I define incorrect. This is part of the instructive element of my lecture. So first I will tell you how it works, which is far from being simple, and it's not understood by most people, partially because they don't spend the 2 days which are needed to understand it. So when you have a chemical reaction, let's say moving a charge from one point to another, you could describe the total energy of the system, including the solvent, by such parabolas which are called Marcus parabola on the name of Rudy Marcus. And the intersection of this parabola give you an estimate of the height of the barrier. Now, in solution, you have to pay a lot of energy to rotate this water. In a protein, you could have the polar groups pointing already in a direction where you don't have to pay too much in rotating the environment. So the parabolas in the protein are closer together. You have smaller amount of this quantity, which is called the reorganization energy, and therefore smaller barrier. I will run it again with you. When you do chemistry in water, and you move charge from one point to another, the water molecule must rotate. This costs a lot of energy in terms of water-water interaction. If you do reaction in enzyme active site, the dipoles of the enzymes are already pointing to the same direction and you don't pay anything. Now, the reason why it is hard to understand it is that the enzyme works fast, not because of the interaction between these dipoles to the charge, but because these dipoles don't have to move when you move from the initial state to the final state or to the transition state. So this is the part that you could read in my papers. This is how enzymes actually work. At least this is how any enzymes that we try turn out to work. Now there are alternative ideas, and all of them could be checked easily by a computer. The earliest idea, which would be trivial to you to understand, though it's incorrect, is that enzymes work by pushing the substrate to a structure which is closer to that of the transition state. When you do a reaction which breaks this carbon-oxygen bond in a sugar, the sugar becomes planar. This is known by quantum mechanics and also by organic chemistry. So the initial idea of D.C. Phillips and others was that the enzyme squish this sugar to a planar form And therefore, it's easier to climb the barrier. It's idea which is called ground state destabilization. It's the easiest idea. When I teach students, this is what I teach them, and then I tell them it is all wrong, because it's intuitively obvious. There is one problem. Enzymes are very flexible. What we just described is like taking one spring and pushing it by many other springs. It's like taking a sponge and taking a pillow and trying to squish it. It would not work. So this one you could easily show by computations that it's not a good idea. Basically, every time when you open biochemistry textbook and see how enzymes work, it's completely incorrect. So I don't have time to show you how incorrect it is, but one of the nicest ideas is that you take a charged ground state and you move it to the enzyme, and the enzyme is an oil drop. So the charge is unhappy. Therefore, it reacts faster. There is one problem with it, that enzymes have nothing to be, they are not oil drop. Essentially, they are extremely polar. So this one also, it's very easy to knock off, but I will just take two ideas that you might like. Two butterfly ideas. One of them, which made Nature and Science many, many times, is the idea that enzyme catalysis is due to distant residues. And the less you understand enzyme, the more you are excited about this idea, because it has the word coupling, distant. It basically makes you comfortable because you feel that neither you nor anybody else understand how it works. And I'm quoting essentially Kissinger on. So there were countless papers along this line, and what we show that actually all of the distant mutations that change the activity of the enzyme, what they actually did, here we show that we reproduce the change in rate constant, they changes the active site reorganization. They changes the dipoles in the active site. And the best way for you to understand it is to look at this picture. You could view enzyme as a nutcracker. It's not the right way, but it's good for this effect. Now, if enzyme wants to be like this, when it is correctly closed, and you made the mutations that open it a little, this structure will change, and the enzyme will react. Now, what is amazing, from people who are occupied with how the brain works, is that none of the people who propose this distance mutation ever want to deal with this trivial explanation, which is much simpler than anything else. Now, here we come to the most popular idea, which require really better tools. Again, the colors are not good. A lot of time people argue that enzyme catalysis is due to dynamics. It's due to the motion of the enzyme active site. Again, it's hard to see here the color, but this idea, if you formulate it correctly, it says that in enzymes we don't have Boltzmann. We have coherent motions. Some physicists like it the most. So when you have open enzyme, when it closes, this motion help it to do the chemistry. And the way, by the way, the first time I heard this idea was in 1976. I came to a seminar in Caltech. And two people who were featured in yesterday morning, I sat near two great physicists. I was just a lowly assistant professor. I just came to the seminar. And an Italian physicist named Carrari explained to who you ever see who they are, that enzymes work by dynamics because he measured enzymes and found that dry enzymes have magnetic properties. And these two people say, great. The two people were Feynman and Gelman, who knew very little about enzyme at this time. And Carrari's explanation has nothing to do with enzyme. He told them that they are magnetic. What magnetic has to do with enzymes? But whenever you throw something and throw in a fact, it sounds convincing. Now, and this is, by the way, what is done here. People show that enzyme, the rate of the chemistry is similar to the rate of opening and closing. Therefore, the process of opening and closing must be responsible for the fast reaction. Now, this would have been correct if enzyme was underdamped, if the friction in enzymes was small. However, the physics of everybody who look on large molecules is that enzymes have very large friction inside, and when you manage to model the enzyme correctly, and I don't think I will have time for it, when you deduce the correct friction of the enzyme, maybe I will have time for this, maybe not, you find that when the enzyme closes, the substrate has to wait near the chemical barrier so long that it forgets anything that it ever knew about the initial motion. Enzyme has no memory whatsoever after it waits long enough before it does chemistry. Again, this is something that at present could be determined only theoretically. And before I'm leaving the enzyme, I would basically, again, try to give you material for thought, maybe also for myself. For the last 30 years, we were predicting correctly the rate of enzymes, the rate of mutation in enzymes, and so on, and knocking down any possible suggestion that anybody suggested. Tunneling, dynamics, compression, everything. I cannot convince you that we prove how enzymes work mathematically, because we've done an experiment. In all of them, we were correct. I cannot do the n+1, which is needed for induction, because it's not an analytical question. However, any opposite proposal could never predict anything. So I would argue that this is a way of understanding, maybe not perfect, but it's a current truth, at least in philosophy of science. OK, so now how much time we have? No, I know, but how much? Because I want to plan my. How much? OK, so now we finish the enzyme mainly as a demonstration of how you ask scientific questions. And now we move to more exotic things, which I think are equally valid. So now, in the case of enzymes, we know to calculate activation barrier. We know a lot of chemistry. It's fine. Now we want to understand motors and more complex systems where they are just more complicated. It's harder to handle them in the computer. And we also want to be able to go to long time scales. So just as a start, when we have a process which occurs very fast, we could model it by direct molecular dynamic simulation. And if this reproduces reality, we know we are OK. So I will show you just one example, a relatively old one. And then we move to the slow reaction. If you want to understand what happened in the first step of the visual process, there is a protein called rhodopsin. It has inside the molecule called 11-cis-retinal. You will see the picture soon. It absorbs light. The protein changes structure. It activates another protein. And eventually, a signal is being sent to the brain. This is the structure of this retinal. This is called cis for you. And when the bond is rotated, it will be called trans. So this is 11-cis-retinal. When we did a simulation, the structure of the protein was not known. But if you assume that the protein is complementary to this site, you could use Newtonian. We heard the good and bad thing on Newton. You could use Newtonian mechanics. It's completely valid, even in the femtosecond time scale, and see how the atoms are moving and try to see what happened to them. So here it's the first simulation of this process, where we started from the excited state where this bond is very happy to rotate, and found that in 100 femtosecond, which was at that time unknown time, this bond rotates from cis to trans and produce a nice movie, which I keep showing all the time. So this shows what happened when you could simulate something with a computer of that time. So this is the retinal in a complementary active site. It gets light, and then it has no objection to rotate around this bond, still try to keep the same structure, and this happened very, very fast. And then it pushes the protein, and the protein will eventually open, not in this simulation, because we did not know the structure of the protein. So this is something that you could do with not so powerful computer, and your simulations will be relatively reliable. Like here is the ground state potential, this is the excited state, or you go to the excited state with light. Eventually, and then you rotate very fast and cross through what is called now conical intersection to the other direction. Anyhow, you could do the same thing in photosynthesis. The question is, what do you do when you have much slower time scales? So now we go to the second or the third part of the lecture, how you model complex systems and how you know that what you do is correct. So we have very large protein. At present, we and others don't have enough power like modeling this ATPase machine, and you still want to see how it works. So the first thing is simplifying the problem. And again, you have to have intuition part of the equation in modeling it. So an early idea was that you replace each side chain by a sphere. And when we used this model in 1975, we were able to take a protein, BPTI, and to fold it near the correct structure. And one of the points which is highly relevant to this meeting is that at that time there was what was called the Leventhal paradox, which said, argue that you cannot, Leventhal knew that protein could fold, but he says that protein have so many degrees of freedoms. How could he find the correct structure? And what was found in this work is that actually the relevant degrees of freedoms are not too many, and it's not a big deal to protein to fall to the right structure. In recent year, we took the same idea but taught the protein to do electrostatics better. Because I told you that we found in enzymes and other systems that the main physics in biophysics is the energy of charges. I wrote about it extensively, but we trained this model to also have correct electrostatic energy, and then I will show you how well it works in 3 cases. So we have a simple model of the protein. It seems to be able to capture the energetics correctly. I will show you later how well it does it. But we also want to teach it to do things in a very long time. So I told you that in short times you could use Newtonian mechanics. When you have long time processes, you use what is called Brownian dynamics. I think yesterday when somebody talked about a modeling of the market or something, it looks to me like Brownian dynamics. The missing part in yesterday's lecture and even here is what type of friction you will use. And we figure out how you get the correct friction. I will say it very fast. You take the real system and apply very strong force to it. Then you take the simplified system with Langevin dynamics and apply the same force. You change the friction until both of them respond to the force at the same speed, and then you know you have the correct friction. And you could use it to model single molecule experiment and so on. And now I'll show you 3 cases where it works. Number 1 is the ATPase that I promised you. So this ATPase has a central shaft that rotates, and when it rotates, it changes this molecule with 3-phosphate to a molecule with 2-phosphate and release energy. Or it go in the opposite and move from the 2 to the 3. Now the remarkable thing is that Kinoshita made this remarkable experiment, single molecule experiment. Oh, let's try to show you. Where he showed, okay. No, this is not what I want to show you, so I'll show you here. You see this guy rotating. This is how the rotation of this motor looks like. Remarkable thing is that it does not rotate as it should, 120 degrees each time. It rotates 80 degrees, stop, does the chemistry, and then rotate the rest. So this is a question that is, I would argue, impossible to explain experimentally. It's possible and ingenious to show it experimentally, but the question why is a question of capturing the energetics. So we use our model, the simplified model. Again, sorry for the terrible colors here. This direction is the rotation, the upper one of the central shaft. This direction is the change of the subunits. So we found that it's actually go to 80 degrees and stop because of electrostatic repulsion, and then it does the chemistry. And with a lot of work that we don't have time to explain, we end up with this map, which actually, if you study it, you will understand how the body uses and produces energy. You will understand it in a logical way rather than in words, because this potential surface, where here you rotate 80 degrees, do chemistry, and release the inorganic phosphate, give you the basis for the action of this motor. If you don't have this map, basically whatever you say on how it works has no logical value because you cannot predict and you cannot explain. So we produce this by computation with a simple model. We also were able to reproduce the time of different steps. And this is just one example. Just to, again, the colors are so bad. One of the things that we show here is the Japanese were really interested, let's put it like this. Every other person explained that this molecule works because of some steric effect, that it stopped at 80 degrees because of some strange repulsion forces. So the Japanese group who worked on it did the following. They removed almost all of this gamma group, and the stuff continued to work as if nothing was done. And we show that if you use the electrostatic model, this is the profile before you did the mutation, this is after. We still have the same character. So we have a model that explains reality without too much problems. And before I go to the last example, we also use the same model to explain how flow of protons from below lead to rotation. And this, how much time we have? Like 10 minutes, 3 minutes? Yeah. So in order to explain this, again, you have to know to do a lot of things. You have to know to model proton transport. You have to model rotation. And we don't have time for it, so I will just tell you that we know to do it. And we end up, again, with a map. And the reason why I show you the map, this direction is the flow of the protons. This direction is the rotation. I will show you later one more map, but I want to make a point. A lot of people discuss on how this coupled proton motion in chemistry work. Essentially, Wolfgang Junger, who essentially was in this room in Marcus' birthday, is the only one who made a logically correct explanation of how the coupling occurs, but even he was unable to formulate it in a way that physicists would be able to agree or disagree. I postulate that unless you have a map, this direction is the rotation, this is the proton, which has the right asymmetry and it comes from the structure, you have not explained how this works. And I will show you one more simple example to make my point really clear. So here I just argue, due to the slowness of my talk, that we could do it. But now I'll show you the nicest example of what one expects from simulation in understanding complex system. And the example I choose is myosin 5. Now, myosin is a family of molecules involved control the muscles, a lot of systems. Myosin 5 is just kind of, it works like this and carry load in the cell. It's the most widely studied myosin system, the best structural information. Countless single molecule experiments in biochemistry. And the nice thing about it, otherwise it will have no value, is that it works in one direction. Now, to impress upon you the problem, this is, if we walk, or this walk, it will be the same if I walk in one direction or opposite, the same speed. This is what we know from daily life. However, this does not happen unless one underwent knee surgery. So there is a question, basic question in the field, or there should be a question, why myosin goes in one direction. And again, you would be surprised how many people summarize it by saying it works in one direction because it works in one direction. This is really the logical context. And we try to do more from the limited structural information. So you build structural models from the available X-ray, electron microscopy, extremely elaborated cycles that I don't want to show you. And what we found is that all the directionality comes just from the fact that when this head has this structure, it is 11 kcal per mole more stable than when it has this structure. So people would, by the way, this is a field where everybody knows all of these names. Rigor, power stroke, essentially, one of the guys that I choose as referee got extremely upset that I don't know the names. But this is mostly what people know. They know to call this stuff names, but it's very hard to know what is the energy of each of these without a computer model. And when you look at this, you find that the only difference in all of this cycle is that here, it's easier to be than here. Now, if you take this knowledge and move to a really complicated diagram, but it's even more complicated in this paper, you find out that moving in this direction has much lower barrier than moving in the opposite direction. The difference in barrier also reproduce single molecule experiment where you try to stop the motion, and it actually explains what is happening. And of course, we could convert it, this is in progress, to a Langevin dynamics, it's not moving, simulation. Okay, what does it say? Media not found. Okay. Anyhow, we have now simulation. We will have more sophisticated one. But the main point is that if you are able to find the source of the directionality, you made a progress in understanding. I could have told you about ion channels and just show you a movie. This is transport of sodium, of potassium through ion channel. These are the chlorine. Again, there are beautiful questions on voltage activation. And I could have told you about the ribosome. And maybe ask me to show this movie. So we also study helicases. Maybe it will work. It needs energy, yes. So we also could explain how this machine pull DNA in. And in summary, from our experience, the key is electrostatic energy. I think that this bunny is known also in Singapore. And at least in this field, the complexity is really the problem, not the solution. And I think we could identify, in many cases, the hidden connection. These are people who contribute on different stages, and thank you very much.
Okay, now it's time for questions.
Now that you have electrostatic model for the cases and for the ATPase, is it feasible to find an analytical solution to describe this with the parameterization which you have now that you have the energetics to look at analytical expression for molecular motor that we can use now?
Yes. So the answer is, I'll just go two more. I think that the answer is that for ATPase, if one take this map and digitize it, which anyhow we did, and then shift it in this way by changing, when you change the concentration of the product and reactant, it's equivalent to moving this map like this. So we do have analytical EVB map to ATPase. There are requirements. If we talk on bed motor, you don't want to go back here. This will be leakage. You see, here we have the ATP. Here we have the ADP. This is what happened in water, a huge barrier, so it does not happen. But if this point was more blue than this point, we will have a leaking motors. So we could use this model, first of all, convert it to analytical, put the concentration, and see what is the efficiency. What we cannot do analytically, we could calculate the effect of mutations on the height of these points, and then suggest how to repair ATPase. If somebody will try to build molecular motor, we could use the same procedure to get the landscape. But we need to move from structure to map, and then we could use analytical simplification. So,
more question?
Please.
Maybe I'll ask a question. So for this myosin,
yes.
You know, this like directional motion.
Yes.
From the starting to the end, that has a thermodynamic driving force. What happened in between? It seems in between is also knows where to go.
Essentially, by the way, I encourage people to ask questions. I will not attack you. It's not so nice to have.
Yes.
One of the interesting things, there are several ways, essentially I have the diagrams below, several ways to get directionality. One of them would have been that the thermodynamics would be different. Essentially, myosin get directionality from having kinetics barrier and not thermodynamics. The ATPase work by having overall a free energy gradient, and then it does not matter what happened. Here, the rate determining barrier is higher in the back direction than in the forward direction. And one of the things which is confusing to many people, essentially, if you want to digitize this and to calculate the kinetics in 2 minutes on your iPhone, what you do, you measure the highest barrier, you measure this difference, and you write Michaelis-Menten, for those who know it, equation, where you know the barrier, which would be like k cut, you know this difference, and you get the result. The intermediate barriers do not count, but the highest one determine the rate.
Okay, thank you.
Yes.
Yeah, maybe?
Yeah.
I have another question. We are very interested in excited state reactions in solar energy conversion.
Yes.
On what are the possibilities to do the computations involving excited ground state
Yes.
reactions here?
Okay, so essentially excited state calculations become a growth industry. There are a lot of people who do this green fluorescence protein, different, I mean, there are people, the difficulty is that if you use a time-dependent DFT, the difficulties are in the quantum approaches, that if you want them to be accurate, they are extremely expensive. And then it's hard to run trajectories. So it's a field which is progressing quite nicely, and it's in the forefront of theoretical chemistry. And things are doable, and there are people who are working on it. Yes?
Okay, Peter?
Thank you for a wonderful talk. I was wondering, so one of the fundamental things that you raise is this issue of multiscale simulation. And we all know that we have this numerical dispersion going on. And so my question would be...
I was hoping to say yes.
Okay, so my question would be, you know, if I go from density functional things all the way up to macromolecule movements, you know, I get lost by definition somewhere on the way. So my question would be, you know, what do you expect the limits of these things are?
Okay, so again, since I've been around for a long time, I heard most of the question, and your question reformulated by David Buckingham many years ago was, we don't know to calculate edge 2. How could you calculate a protein? The answer is very simple. We are not trying to, there are two types, because I also have a way, which I call paradynamics, to get even accurate result. But what we do most of the time, we don't invent quantum mechanics. We take the properties of the reference molecule in solution from experiments and very high-level calculations, and we only calculate the change in energy moving from one phase to another, where we know exactly what is our error, so we have a complete confidence.
Well, but there's a danger there, right? So if I would do if I would take a three-dimensional structure of an enzyme, and I would say, okay, I cannot calculate the thing from a priori, so I have to start with some kind of confirmation, and then only look at the changes in that confirmation, I might have a wrong starting point.
No. Though we did work, or we are working on folding, what we do, we start from known enzyme active site. The changes in the active site when you move to the transition state are very small, and when we do our free energy calculations, we have full control on the convergence. So we know how much changes we could handle. We have, let's say, 30-year experience with this. So it's not, we even done calculation with large structural changes and done very well, but we never trying to predict the structure from scratch and then to have the catalysis. So we always keep at least one leg on a safe ground. And if we had more time, I would show you how we also essentially, why not? We always end up with people who say that, well, we don't believe this EVB, it's a kindergarten approach. So we have to show them that we actually know to do things very accurately. Hey. What happened? Okay, I think this was a hint. What we do a perturbation from a simple potential to ab initio potential. We do it at the initial point and the final point. So instead of calculating the whole free energy on very high level ab initio, we calculate the free energy on the simple potential. And then we do perturbation from the simple potential to the ab initio, where we could go even to couple cluster. So we always spend time showing, this will, anyhow. So we spend a lot of time proving the critics. First, we do things which people claim is ad hoc. And then we have to spend a lot of time showing that it's also rigorous.
But if I'm allowed.
Yes.
So, but in the end, what you need to do is you solve a thing, let's say, on one level. Then you make some kind of abstraction, and then you put that, feed that into the next level, so to say, because you cannot do all those spatial time scales at the same time. So that means that there are approximations in there for the starting point of the next level.
I would say no, but I'm not sure I could articulate. It goes like this. When we have several recent papers which show this paradynamic approach, and they go like this. We take medium level potential and calculate the free energy, medium level ab initio, calculate the free energy of moving from reactant to product on this potential. Then we show that if we use our approach of using a reference potential, we get exactly the same result. So we never have, whatever we do, we never have analytical absolute proof, but we always have a numerical proof, and we do it again and again. So since we do a perturbation approach, we could also do it in many steps, and if we get the same result, it means it's a correct approach, or at least in my view. Yes.
Yes.
Wait, let's resuscitate this one.
This walking process, have you incorporated a learning function in the way it walks? Zero.
You see, one thing that is taboo for me, and okay for everybody else, everything is it must be physically based. It must be based on the potential surface. We don't tell him how to walk. It comes from converting the structure to energy. There is nothing, we did not tell him, this is why it is convincing to me. We don't tell you which direction to go. And then we find you go in the right direction. So probably we've done something correctly.
But that means that there is an algorithm which says the lowest energy, I go in that direction.
No. When I say no, it's always yes and no. It goes, it goes to the right direction because of the energy. But we are not telling him which direction is the lowest energy. It comes by taking the structure and calculating the energy and finding that this structure has lower energy than this structure. You see, we are not God. We did not build myosin. It's in the protein sequence. So if we take the structure of the protein like this and the structure of the protein like this and calculate the electrostatic energy, we find that this one is 11 kilocalories more stable than this one. From this conclusion alone, we reproduce directionality and everything.
Thanks very much for your wonderful talk. I have questions because I'm a PhD student from School of Computer Engineering. I'm curious about how do you have such kind of idea using simulation to do research in chemistries, and how do you convince people when has your first kind of ideas?
No, first of all, you don't convince people.
Because
there are two schools. One is Niels Bohr was asked how he convinced all of these people in quantum mechanics, and he said, I never convinced them, I just outlived them. So you don't... No, you do it, and you keep seeing that the results make sense, and then other people are doing it, and this is how science works.
And how do you validate your result?
This was the whole part of my talk to this community. No, really, it's a good question. You validate it by seeing that every time when you choose another problem, you get what people observe experimentally. Sometimes, like in the case of this vision, you find the answer before the experiment is done. So you validate it. This is what I call induction, though it's rigorously not correct. If every time when you do a calculation, you get a number which agree with experiment, and if people who take the program get similar results, you convince some people.
And another question is that behind your simulation, there's a lot of models you propose. And how do you think using, for example, currently there's a lot of big data. Is it possible to combine your model and big data to?
No, this is orthogonal. Not that I'm sure what is big data, but I hear it a lot. This model is, there are two directions to go. One is informatic, to incorporate everything that's known. The other is physically based model. A similar thing in protein folding. At present, people who use informations are folding better proteins than those who are using physically based models. So you don't, the only place where you use the data is to improve the parameters for the forces in your model. But you are not allowed to tell your protein how to fold. It should find it by itself. It's a different scientific philosophy, not necessarily better one, but it's different.
Thanks very much.
No?
If you want to show the slide later, now it's working.
Which slide I forgot? By the way, it's not working, it's rotating. No, but I want this one. These young people are so smart. Yes. Okay, so this approach, which we have several recent papers, including very rigorous one, and we chose the name paradynamics just to tease the metadynamics community. We calculate on one potential where we could have very fast calculations. We calculate the free energy. And then we do perturbations to, let's say, the correct potential, which could be ab initio. And we have extra ingredient. We also have an approach which changes this potential until it becomes as similar as possible to the quantum potential. And with this approach, we went to quite high level ab initio. And we did the whole cycle, because in some cases, we also calculate this stuff. And it's the way to show people that by the way, you always get the same result as the EVB, but it's to convince people that this is what you get. And maybe this approach we use also for green fluorescence protein to calculate the color. So you use simple potential, namely QCFF pi, and then you do perturbation to time-dependent DFT.